Hello again, everybody. Mr. Umler here. Welcome back to today's lecture, which is going to be about cell membrane function. As you recall, our last lecture was about cell membrane structure and all the different components that are found in a cell membrane that give it this quality of being semi-permeable or selectively permeable. So just things to keep in mind, all the different parts of the membrane we talked about last time that are going to contribute to this. We have our phospholipids, which remember they have that hydrophilic head with the hydrophobic tails. We also had the proteins. We're going to talk a lot about those today and how they regulate the permeability of a cell membrane. And then there's also the carbohydrates, which we're not going to talk about really today. Those are going to come in a little bit later in this unit when we get to cellular communication and things like that. But some of the terms we are going to be talking about today are passive transport, which are forms of transport that require no energy input. So what that means is the cell is not going to expend any energy in making these things move across the membrane. As opposed to active transport, which as the name kind of implies, these require energy in the form of ATP. And we'll be talking a lot more about how cells use that ATP in order to move things across them. The first thing we want to talk about, though, to give you an understanding of why things want to move, is this idea of something called the concentration gradient. And the concentration gradient is basically just a comparison between the concentration of something in one area versus the concentration in an adjacent area. And you'll notice here on the right, there's a very high concentration of these blue dots, and on the left, there's a very low concentration. And all things being equal, molecules or anything for that matter is going to want to move from high to low concentration. Imagine you're, in a room, you're out at a restaurant or something like that, and there's 50 tables in the restaurant. You don't want everybody to be sitting right next to each other. You want everybody to be nice and spread out. Same thing in the classroom. We don't want everybody clumped in one corner. People are going to be uncomfortable. They're going to want to move to where it's a much lower concentration. And generally speaking, when you're going from high to low, that's going to be passive transport. Those are going to be the things that don't require energy. If we're going from low to high, those are going to be the active transport mechanisms that you're basically making things go where they don't want to go. And so another way to think of this is concentration gradient is kind of like a hill. So up here at the top of the hill, this is our high concentration, and this is our low. And generally speaking, things want to move from high to low. So without any sort of energy input, those things are just going to spread out, and then our ball is going to roll from the high part of the hill down to the low. In active transport, we're going to go the opposite way. Things are going to go from being spread out to being close together, or they're going to be moving up that hill. And again, that's going to require some ATP, some energy to make that happen. So first we're going to start off with passive transport and the various modes of passive transport. The first one of these is simple diffusion. And the definition of diffusion is basically movement from an area of high concentration to low. And so as you'll see here in this animation, all these little dots down here, they're moving. They start off in high concentrated area, and they're going to move to a more low concentrated area up here towards the top. And this is basically influenced by three determining factors. Three things are going to affect the rate at which things diffuse. And the first of those is diameter. So bigger objects are going to diffuse more slowly. Smaller objects are going to diffuse more quickly. Along with that, we have temperature. So temperature, high temperature is going to cause a more rapid diffusion. Low temperature is going to cause a slower diffusion. This is really easy to see if you've ever noticed the difference between trying to stir sugar into a cup of hot tea. You put that sugar in there, you stir it up, and it dissolves really quickly. You put sugar into iced tea, and you stir it up, and it takes a lot longer to dissolve because the molecules are moving much more slowly. And then the last thing that's going to determine the speed of diffusion is the gradient. So again, think of that hill from the previous slide. So if you have a gradient that's represented by this triangle, diffusion is going to happen at a reasonable rate. But if you have a much steeper gradient, diffusion is going to happen much more quickly. So again, think of that ball at the top of the hill. It's going to roll down that hill much more quickly than it's going to roll down this hill. Anybody who's ridden a skateboard or a bicycle knows that the steeper the hill, the faster you get down to the bottom. Also, diffusion that happens within cells, so imagine this was a cell right here, is generally going to happen pretty quickly because there's not a whole lot of space inside of this cell for things to move. Diffusion across the membrane, however, so from the outside here to the inside, that's going to be really dependent upon these things. Mainly the diameter and the gradient, because when we're talking about living things like us, for example, the temperature is going to remain fairly constant. But the diameter of the molecules and the gradient definitely are things that can change. All right, so the next type of passive transport is what we call osmosis. And this is another form of diffusion, but this is diffusion specifically of water. And the way that you can tell which way that water is going to move, there's three terms that you really need to be familiar with. So the first of those is hypertonic. The next is hypotonic. And the last is isotonic. So the first thing about these terms is that you're always going to be comparing two things together. You know, it's inappropriate to say that a solution is hypertonic by itself. What's more appropriate is to say that it is hypertonic in comparison to some other solution. It's kind of like looking at a person and just saying, oh, well, he's taller. It doesn't really tell you much unless you know who he's taller than. If you say he's taller than me, that's not really saying a whole lot. I'm not very tall. But if you say he's taller than, say, Kareem Abdul-Jabbar, that's saying something. He's a lot taller than I am, and that's significant. So the tonicity of a particular substance refers to how much stuff is dissolved in it. So a hypertonic solution has more dissolved solutes, whereas a hypotonic solution is going to have less dissolved solutes. And an isotonic solution has the same amount of solutes. And this is really significant when it comes to living cells and how water is going to move in and out of those cells. So let's look at a few examples of that. So over here, we have an example of a solution that is hypertonic compared to the cell that's in it. So our solution, whoops, sorry about that. Our solution is hypertonic, which means the cell is hypotonic. And what that means is water always wants to go from an area of high concentration to low concentration, just like anything else. So if you have a lot of dissolved solutes, a hypertonic solution, that's the same as having a low concentration of water. If you have a hypotonic solution, that's the same as having a high concentration of water. So the water is always going to flow from the hypotonic solution to the hypertonic solution. Now, if any of you have ever gone to the hospital and had an IV put in you, you may have noticed that the saline solution has a label on it that says 0.9% normal saline. And that's significant because that is the amount of salt that's in our blood. If you took a cell and put it into a solution that was, say, 2.5% saline, now that solution is hypertonic and your red blood cell is hypotonic, and that water is going to flow out of the cell into the solution, and those red blood cells are going to shrivel up, which is not good. Conversely, if you had a solution that was 0.0% saline, now our cell is hypertonic, and the solution is hypotonic. And again, the water is going to flow from hypo to hypertonic, which means that the water is going to flow into our red blood cell and it's going to swell till it actually bursts. So this 0.9% solution, that's right here. This is our isotonic solution. So a misconception that a lot of people make here is that there's no water moving at all. But actually what's happening is the water's moving out and moving in at the same rate. For plants, they have something called a cell wall, which our cells don't. So plants actually like to be in a hypotonic solution. That's going to cause water to flow into the cells. They're going to swell. They're going to push up against that cell wall. And that's going to create something called turgor pressure. And so a plant that has lots of water in it is going to be nice and rigid. It's going to stand up tall. Plants that have less water in them, or you put them in salt water, they're going to squeeze that water out. There's not going to be that turgor pressure against the cell wall, and they're going to go limp. All right, our next form of passive transport is facilitated diffusion. So again, this is diffusion, but it's specifically diffusion through a protein. So there's a couple different kinds of proteins that are involved in this. There's what we call channel proteins, and exactly as the name implies, they're just an opening or a channel for molecules to pass through. So molecules diffuse through an open channel. As opposed to carrier proteins, which actually bind to whatever's moving through them and facilitates a much more quick diffusion across that membrane. And there's a specific channel protein that's really worth mentioning for us, and these are called aquaporins. And these are the channel proteins that water is allowed to diffuse in and out of the cell through. So aquaporins are always going to be open. They're always going to provide a way for the cell to get water in or get it out for that matter. There are other types of channel proteins. There's what we call gated channels. And there are ion channels. And these channels actually have kind of a trap door that will close over them. And gated channels will open and respond to a ligand, which is a chemical signal. And ion channels will open in the presence of a particular ion. And we'll talk more about those as we go a little bit further on in this unit. So the other main type of transport that cells are going to use is what we call active transport. And so again, this is the one that requires ATP. This is one that requires energy. Generally, it requires energy because now we're either moving things from low concentration to high. So again, we've got that concentration gradient, and our ball is rolling up the hill. So that's going to take energy. The other reason why it might take energy is because we're moving extremely large molecules. And a lot of times that involves the entire cell changing shape in order to fit these molecules in. So the first type of active transport I would like to talk about is just using, these are kind of referred to as molecular pumps, because we're pumping materials from low concentration to high. And this shows you three different types of these pumps. The first one here on the left is what we call a uniporter. It's moving one single thing in one direction. The next one is called a symporter. And now we've got two different molecules that are both being moved in the same direction. And the last one is called an antiporter. And we're moving two different molecules in two different directions. And the most common example of this are what we call these sodium-potassium pumps. And these generally move sodium into cells and potassium out of the cells. And this is one of the most common membrane channel pumps that you'll see, and they're found in almost every type of cell in every living organism. The next method of moving materials, this is now when we're talking about moving something really large into a cell. And this is what we call endocytosis. So endo meaning taking into, cytosis, we're talking about cellular materials. So here you see this large molecule that needs to be brought in. It approaches the surface of the cell membrane starts changing its shape, and it actually engulfs, encircles, and creates a vesicle that particle is now inside of. There's two versions of endocytosis. The first is phagocytosis, and this is for very large particles. Phagocytosis literally means cell eating. And then the other is called pinocytosis, and this is for smaller things that are still too big to just squeeze between that membrane. And pinocytosis literally means cell drinking. So phagocytosis is cell eating, pinocytosis is cell drinking. Another form of endocytosis is what we call receptor mediated endocytosis. And this is basically the same process, but it's just much more specific. So you'll see these little blue dots on the surface here. These are receptors, and they have, there's kind of this little Y-shaped piece, and the blue dot is the ligand, or the chemical that's binding to that receptor. And so these specific receptors are going to detect the presence of that, and it's going to form, again, a little vesicle that's going to take now these materials into the cell. And that's exactly what you're looking at here. This is an actual electron microscope picture. You've got all these molecules that have bound to the receptors. They create this infolding of the membrane, and then it eventually splits off, and now you see all that stuff is now inside of this vesicle inside the cell. And lastly, when cells want to get rid of large amounts of material, they use something called exocytosis. So exo meaning out, cytosis referring to the cell. It's a way of getting things out of the cell. So imagine this vesicle right here. Maybe this is filled with proteins, little proteins that were just made in the endoplasmic reticulum. They've been packaged in the Golgi, and this vesicle now makes its way to the surface of the cell. And this membrane that binds the vesicle actually binds to the cell membrane. It opens up and releases those contents to the outside. This is a great way to release hormones. It's a good way for cells to get rid of waste products that they don't need anymore, or just to release proteins that are needed in other areas of the body. They'll be secreted from the cell this way. And then this membrane that was part of the vesicle actually becomes part of the cell membrane itself and allows the cells to get bigger until they ultimately need to divide. So those are the main forms of cellular transport. Passive transport, again, is the transport without energy requirements from the cell, and active transport is the transport that does require energy from the cell, either because things are being moved from low concentration to high, or because we're moving large amounts of materials into or out of the cell. So hope that helped, and we'll see you next time.